Hi everyone, my name is Chantal Claassen-Bates and I will be presenting Module 3 of the 16S Intermediate Bioinformatics online course for 2019. I would highly recommend that you first cover the three-part series for Module 2 before starting with Module 3. The primary outcome of this module would be for you to be able to describe different aspects of planning for 16S rRNA experiments, for example, study design, DNA extraction methods, and laboratory workflows. We'll be focusing on the 16S rRNA gene and why we are interested in this gene. We'll touch on concepts behind Sanger sequencing and high-throughput sequencing. We'll look into why it is important to clearly plan an experiment and how different components to the study and experimental design may influence data generated and downstream analysis. This module will also serve as an introduction to some techniques other than 16S rRNA sequencing and why they are of importance. As for Module 2, I've decided to do a couple of recordings on the topic of sample collection, DNA extraction, and library preparation for 16S NGS analysis by splitting it into different parts. In part 3.1, I will be focusing on describing the 16S rRNA gene in a bit more detail. So, how do we study our microbes? The first microbes were described in the mid-1600s. It was during the 1800s that connections were made between microbes and disease, and ever since, culture, staining, and microscopy have been used to study microorganisms. A huge advantage to, for example, culturing, especially during disease associations, is that only viable organisms can be cultivated. One of the primary disadvantages, however, is that previously it was thought that less than 1% of bacterial species are cultivatable. Hence the rise of molecular techniques such as Sanger sequencing in the late 1900s. So let's take a closer look at some of these culture-independent methods. There are marker gene-dependent and marker gene-independent approaches. Marker gene-independent approaches, where we use high-throughput sequencing to perform what we call shotgun sequencing, is where DNA is extracted from a specimen and sequenced without PCR amplification of a marker gene. So essentially, you will be generating all microbial information, may it be bacterial, viral, fungal, etc., from a specimen. The major limitation to this technique, of course, is its cost. Marker gene-dependent approaches is where we use, for example, fingerprinting techniques, Sanger sequencing, or high-throughput sequencing to sequence a specific marker gene which have been targeted with a PCR step. For bacterial studies, we are mostly interested in the 16S rRNA gene due to three primary characteristics. Firstly, it's present in almost all bacteria. Secondly, the 16S rRNA gene function has not changed over time, which suggests that random changes in sequences are a more precise measure of evolution. And thirdly, the 16S rRNA gene, which is approximately 1,500 base pairs in length, is large enough for informatics purposes. With targeted 16S rRNA gene sequencing, however, we can only determine the presence or abundance of bacteria, whilst shotgun metagenomic sequencing will also reveal the associated metabolic pathways. So how do we use the 16S rRNA gene in experiments? The 16S rRNA gene is approximately 1,500 base pairs in length. This plot is representing the percentage of bacteria with the same DNA present at each position of the 16S rRNA gene. So the larger the percentage, the more organisms have this nucleotide at this position. The part of the gene which are structurally important for the ribosome to work are referred to as conserved regions, also shown here in grey. Other parts of the gene have less overlap between different organisms due to mutations, but still allow the ribosome to work. These regions, shown in different colors, are the variable regions of the 16S rRNA gene. Except for the fact that improvement in technology is now allowing for sequencing of the entire 16S rRNA gene, previously it has been one of the limitations for targeted 16S rRNA sequencing. So most studies to date have identified 1, 2, or 3 variable regions from the 9 hypervariable regions of the 16S rRNA gene for sequencing. Primers are designed to anneal to the conserved regions, allowing for amplification and ultimately sequencing of the variable region of interest. Stool bacterial profiling studies have, for example, primarily been targeting the V4 region. So in short, steps during a 16S rRNA gene-dependent approach would be to extract bacterial DNA from the specimen under study, amplify the preferred hypervariable region or regions of the 16S rRNA gene, sequence the amplicons to determine which bacteria are present in a sample. This technique also provides information on the relative abundances of bacteria detected from a sample. And we can even determine how related these bacteria are to one another. But before we have a look at how the technology works for high-throughput sequencing, I'll first explain the process of traditional Sanger sequencing. Sanger sequencing is a highly accurate method to basically read sequences in an amplified DNA fragment base by base. One way of performing Sanger sequencing is to cultivate the organism of interest, extract DNA from a single colony, and amplify the gene of interest using PCR. The first step is for the double-stranded DNA to be denatured using heat. Once the double-stranded DNA has denatured, the temperature is lowered. The primer attaches to the DNA template strand. The DNA polymerase then starts adding nucleotides according to the sequence of the complementary nucleotides on the DNA template strand. DNA synthesis stops when the DNA polymerase adds a dideoxyribonucleic acid. Synthesis stops at that position and the DNA polymerase falls off. DNA polymerase does not discriminate between the dideoxyribonucleic acid and the deoxyribonucleic acid, and this results in new DNA strands of varying lengths. Synthesized DNA strands from each of the tubes containing the 4 dideoxyribonucleic acids DDCTP, DDTTP, DDGTP, and DDATP are loaded into separate vials of a polyacrylamide gel. DNA strands are separated by electrophoresis with the precision of a single nucleotide. The radioactively labeled base shows on an X-ray of the gel, and the gel is read from the bottom, starting with the shortest fragment to the top, finishing with the largest fragment. So just to recap, before we look at the technology behind high-throughput 16S sequencing, Sanger sequencing of the 16S rRNA gene is challenging in complex polymicrobial samples. Hence, it requires culture steps. High-throughput sequencing of the 16S rRNA gene, on the other hand, allows for identification as well as relative abundance quantification of all bacteria present in a sample by massive parallel sequencing. In addition, it allows for sequencing of multiple samples at the same time. Here I'm explaining the process of massively parallel sequencing by synthesis. During the process of amplification, each sample is indexed with a unique barcode which allows for multiplexing of samples. Following amplification of the target region of the 16S rRNA gene, barcoded PCR products are pooled at equimolar concentrations, and the library is further processed for sequencing. Fragments are hybridized to the dense lawn of oligos on the flow cell. Copies of the fragments are then bound to the flow cell surface. Clonal bridge amplification results in hundreds of millions of unique clusters. This is followed by cleaving of the reverse strands. The sequencing primer is hybridized to the DNA templates. For each cycle, fluorescently tagged nucleotides competes for adherence to the growing chain. Only one fluorescently tagged nucleotide is incorporated in the complement strand based on the nucleotide sequence of the template strand. This is followed by a fluorescent signal being emitted, which allows for base calling. Hundreds and millions of clusters are sequenced in a massively parallel process. Raw sequences can then be separated based on the unique barcodes incorporated for each sample. So in summary, 16S rRNA gene is by far the most common housekeeping gene targeted to study bacterial phylogeny and classification. High-throughput sequencing targeting the 16S rRNA gene allows for large quantities of DNA to be sequenced much more quickly and cheaply compared to Sanger sequencing. High-throughput sequencing of the 16S rRNA gene allows for identification as well as relative abundance quantification of all bacteria present in a sample. The 16S rRNA high-throughput sequencing allows for processing of multiple samples in a single reaction via the use of bar-coded primers, also referred to as indexed primers. So that's all for part 3.1 of Module 3. Please stay tuned for part 3.2, which will be focusing on study design and sample collection.